So, welcome to session 3 um, of the chapter Human Health and Diseases. So, in this session, we will be seeing about immunity. Every day we are exposed to large number of infectious agents. However, only few of these exposures result in disease. Why? This is due to the fact that the body is able to defend itself from most of these foreign agents. This overall ability of the host to fight the disease causing organism conferred by the immune system is called as immunity. So, immunity is of two types: innate immunity and acquired immunity. So, we will be seeing about innate immunity and acquired immunity in part 1. Innate immunity. Innate immunity is non-specific type of defense that is present at the time of birth. This is accomplished by providing different types of barriers to the entry of the foreign agents into our body. Innate immunity consists of 4 types of barriers. These are physical barriers. Skin on our body is the main barrier which prevents entry of the microorganisms. Mucus coating of the epithelium. So, repeating it again, innate immunity is a non-specific type of defense that is present at the time of birth. This is accomplished by providing different types of barriers to the entry of foreign agents to our body. Innate immunity consists of 4 types of physical barriers. These are physical barriers, physiological barriers, cellular barriers, and cytokine barriers. Physical barriers. Skin on our body is the main barrier which prevents the entry of microorganisms. So, skin on our body is the main barrier which prevents the entry of the microorganisms. Mucus coating of the epithelium lining the respiratory, gastrointestinal, and urinogenital tracts also help in trapping the microbes entering our body. Respiratory, gastrointestinal is basically the digestive kind of system. Gastro is stomach, and then urogenital is basically the reproductive and the urinary. And the excretory system. Okay. So now, physiological barriers. Acid in the stomach, saliva in the mouth, tears from our eyes all prevent microbial growth. Yeah, all these are like chemical stuff, right? Acid in stomach, saliva in the mouth, and tears from the eyes. Now, cellular barriers. Certain types of leukocytes, such as the WBC of our body, like polymorphonuclear leukocytes, PMNL, neutrophils, and monocytes, and natural. Okay, so WBCs of bodies like PMNL, neutrophils, and monocytes are natural killer type of lymphocytes, and natural killer type of lymphocyte in the body, as well as macrophages in tissues, can phagocytose and destroy microbes. So what they mean is PMNL, the neutrophils and monocytes, and the natural killer type of lymphocyte in the blood, as well as the macrophages in tissues, can phagocytose and destroy microbes. So neutrophils and monocytes are one group, and then lymphocytes and macrophages are other groups. Okay, they have similar functioning. Now, cytokine bacteria. Virus infected cells secrete proteins called the interferons. They protect the non-infected cells from further viral infections. So, some cells that are infected by the virus, they secrete proteins known as interferons. Very, very important. We'll also be seeing about alpha interferons. Okay, the interferons are basically protein. Okay, and these protect the non-infected cells from further viral infection. Okay, now moving on to acquired immunity. So, acquired immunity, on the other hand, is a pathogen specific. It is characterized by memory. This means when our body encounters a pathogen for the first time, it produces a response called primary response, which is of low intensity. Subsequent encounters with the same pathogen elicits a highly intensified secondary or anamnestic response. Very important. This is ascribed to the fact that our body appears to have memory to the first encounter. So the primary and the secondary immune responses are carried out with the help of two special types of lymphocytes present in our body, such as the B lymphocyte and T lymphocytes. So we have always known about our immune system. We also learned about the WBCs, and we learned about lymphocytes over there. We know B lymphocytes and T lymphocytes, bone marrow, thymus, right? So the B lymphocytes produce an army of proteins in response to the pathogens in our blood to fight with them. These proteins are called as the antibiotics. What does the B lymphocyte do? It produces an army of proteins in response to the pathogens in our blood to fight with them. So these are called as the antibodies. And the T cells themselves do not secrete antibodies, but they help B cells to produce them. What do? They help the B cells produce them. Each antibody molecules have 4 peptide chains. Okay, so 2 small called light chains and 2 longer called heavy chains. So T cells, they help the B cells. Okay, so now the B cells, it secretes antibodies, as we know. Antibodies act against the antigens. So now these antibodies, they have 4 peptide chains. They have 4 peptide chains, 2 small called the light chains and 2 longer called the heavy chains. Hence an antibody is represented as H2L2, heavy light. Different types of antibodies are present in our body. IgA, IgM, IgE, IgG are some of them. Okay, so a cartoon of an antibody is given in figure 8.4. Because these antibodies are found in the blood, the response is also called as humoral immune response. Humoral immune response. This is one of the 2 types of our acquired immune response, antibody mediated. The second type is called the cell body mediated immune response. So humoral response is the acquired immune response, that is antibody mediated, and the other one is the cell mediated immune response or the cell mediated immunity, CMI. The T lymphocytes mediate the CMI. Very often some human organs like heart, eye, liver, kidney fail to function satisfactorily. So transplantation is the only remedy to enable the patient to live in a normal life. Then a search begins to find a very suitable donor. Why is that the organs cannot be taken from just anybody? We know that there are some reactions that take place. They find it to be foreign body. So there must be a match, right? So what is that the doctors check? So of course blood group is there, all those are there. There must be a match, right? Grafts from any source, an animal, another primate, or any human being cannot be made, since grafts would be rejected sooner or later. So tissue matching, blood group matching are essential before undertaking any graft or transplant. And even after this, the patient has to take immunosuppressants all his or her life. So the body is able to differentiate self and non-self, and the cell-mediated immune response is responsible for their graft rejection. So what does these T cells do? They differentiate self and non-self, and cell-mediated immune response is responsible for the graft rejection. So we already saw that CMI, right? So they have a cell mediated immune response. So now what happens is that this is the structure of an antibody molecule. You can see the heavy chain over here, you can see the light chain over here, you can see that what heavy chain, heavy chain, light chain, and this is the antigen binding site, antigen binding site, right? So S, you have S all over here. Let me find out the meaning later. So this is the structure of an antibody molecule. So it binds the antigen and it does not allow the antigen to act. Okay, so that's all for part 1, session 3. So in this session we just learned about innate immunity, physical barrier, physiological barrier, cellular barrier, and the cytokine barrier. And we also learned about the acquired immunity. We learned about B lymphocyte primary response, secondary or anamnestic response, B lymphocyte, T lymphocyte, then light chain, heavy chain, H2L2, IgA, IgM, IgE, IgG, and we also learned about humoral immune response, antibody mediated, cell mediated immunity, cell mediated immune response, CMI, and we also learned that T lymphocyte is the one that mediates the CMI. Okay, and we also learned about grafts and self and non-self differentiation. Thank you.